So we've talked previously about analyzing the primary sequence of amino acids inside of a protein, but at the HL level, you also have to understand a technique that gets used to figure out things about the concentration of protein in a given sample. And there is also a technique to understand the tertiary structure of a protein, which we mentioned in the core, part of topic 21. That's X-ray crystallography, and X-ray crystallography has been really important for understanding and figuring out what the three-dimensional structures of proteins are at their active site and just throughout. And as computer modeling gets better, that's pretty important for understanding how enzymes actually work. But it doesn't appear to be part of the biochemistry topic anymore. But what is part of the biochemistry topic now is this technique known as UV visible spectroscopy, or UV-vis, it may be abbreviated as. So a solution of protein can be placed in a spectrometer or spectrophotometer, a colorimeter, something along those lines, and you pass light through it. So it could be UV and visible, or just one of those, shown through the sample, and the exact absorbance of that sample can be used to identify proteins, because it can be unique for each protein, but then it can also be used to figure out how much protein is in the sample based on how much of the light gets absorbed and how much gets through. So some proteins are going to contain pigment molecules. Pigment molecules, as we'll go into in more depth later, are things that give molecules color. And so things like heme groups inside of hemoglobin are pigments. It's one of the reasons why blood has a color. So pigment molecules will absorb visible light and therefore appear colored because you're going to see the complementary color to whatever's being absorbed, just like back in the transition metal topic. But most proteins, the majority of proteins, are colorless because they don't have pigment molecules in them, and so they don't absorb visible light. So these proteins absorb ultraviolet light, and so they can produce a characteristic spectrum in the ultraviolet portion of the spectrum. Right here, I just thought it'd be interesting to look at the two different spectrums of blood with and without oxygen. So if you're looking at this spectrum, this is hemoglobin bound to oxygen and hemoglobin not bound to oxygen. You can see how the types of light that it absorbs, because of that bound on oxygen, that oxygen essentially acts like a ligand with the iron that's inside the pigment part of hemoglobin, and it changes the type of light that gets absorbed. And let's see if you can figure out, using the color wheel and the wavelengths of light that are being absorbed, what is going to be the color of oxygenated blood versus deoxygenated blood. Can you try to figure that out? See if you can do that before class. Of more emphasis in the HL topic, however, though, not figuring out the spectrum, but just figuring out the concentration of protein. So although proteins are colorless, there are a lot of solutions that you can add to proteins that will bind with the protein and create a color. Usually these solutions contain a transition metal ion, and so the protein ends up binding to the transition metal ion as a ligand, and that is going to change the color of the solution. So one example is this reagent, Biuret reagent, and it has copper 2 ions in an alkaline solution. And if you add protein to it, it will become more and more purple. You do not need to remember this specific reagent. It's just an example. As we've talked about before in class, I know I've shown you that you can use a spectrometer or a colorimeter, and you can measure the absorbance of light, a particular wavelength of light, and how that changes with the concentration of the solution. And we saw that if you graph this, as concentration goes up, absorbance goes up in a linear fashion for any given wavelength of light. And so this equation's in the data booklet. It's only for the HL biochemistry topic. The symbols in it are that you've got the initial light intensity before it goes through the sample, you've got the intensity after it comes out of the sample, and the combination, all these things together, stand for absorbance. Essentially tells you what percentage of light is getting absorbed by the sample. These other symbols are, so molar absorptivity is going to be different for each substance. The path length of the solution is how wide the actual physical sample is, and C is concentration, which is really what we're going to care about. Now, if you think about it, you can think about this equation, if you make it nice and linear, is that this whole thing here, log of the two different intensities, can be thought of as y. And if you're not changing these two values, what the solution is, or how big the physical container is, then these two things are constant, and that's going to end up being the slope of your line, of your trend line. And so you have this nice linear relationship between concentration here as x and absorbance here as y. And so there shouldn't be a B value, because if the concentration is 0, it shouldn't absorb any light. So that's why there's no plus B at the end of the equation. And it's pretty straightforward. If you create a calibration curve using a bunch of known concentrations and absorption, you can create a nice linear line, get the equation of that trend line, and then you can test an unknown, which will give you an absorbance value. You can then use the trend line equation, or the trend line itself, to work backwards and figure out the unknown concentration of protein. So this is a fairly standard technique to figure out unknown concentrations when things have color, because concentration and how dark something is will have a nice linear relationship between concentration and absorbance of light. And so this is just a technique to measure and determine the concentration of proteins in unknown samples.